Hello. So we have four main topics. You can see here this is a product for protein that is in the market, and you can see how the lab supervisor prepare a purification column to separate a commercial biological product. So for the first topic, subtopic, proteins as a biotechnology product. Proteins is a large molecules, of course, will go fast through that required for structure, function, and regulation of living organisms. In 2000, NIH launched the Protein Structure Initiative, and it's an effort to identify the structure of human proteins. The goal is to able to model unknown protein structure based on structure comparison to those stored in the database. More than 1200 superfamilies of protein are identified so far. The relationship between proteins amino acid sequence to its structure is well understood, and there is a public database hold more than 33,000 protein sequences. The recombinant DNA technology made it possible to produce a specific protein on demand, like enzymes, hormones, and antibodies. And this table is for knowledge. You can see the some enzymes and their industrial applications. This table as well for knowledge, you can see some protein-based pharmaceutical product. Most of them are produced as recombinant proteins. These biotechnology drugs and other medical application produced through microbial fermentation or mammalian cell culture. These can produce large batches of protein products in a bioreactor by growing the host cells that have been transformed to contain the therapeutic gene of interest. The cells are then stimulated to produce the target proteins through precise culture conditions that include balance of temperature, oxygen, acidity, and other variables. At appropriate time, the proteins are isolated from the culture, tested at every step of purification, and formulated into pharmaceutical active product. The second subtopic is protein structure, and of course most of you are familiar with that. You have a primary protein structure that is like B-shaped, like the first figure, and due to the formation of the bonds between the hydrophobic amino acids and the alpha helix and beta sheets, a secondary structure appear, which is the alpha helix and beta sheet. These alpha helix and beta sheets soon come into three-dimensional polypeptide chain and are formed when these secondary structures are close linked, and once reaching this level of complexity, the tertiary structure, these determine the protein function. Then you have the quaternary structure, which is unique for each protein, and it's globular, and it's a three-dimensional complex built of several polypeptide, not only one polypeptide. And here you can see a structure of a protein complex that consists of four polypeptides. Protein folding. The structure and function of a protein depends on protein folding. If a protein folded incorrectly, so its desired function of protein is lost, and misfolding protein are can be degraded inside the cell. In 1951, the alpha helix and beta sheets were discovered. Their structure are very fragile, and the incorrectly folded protein can lead to disease, including Alzheimer, cystic fibrosis, and mad cow disease, and other forms of cancer, and even some heart diseases. A challenge of biotechnology is to understand and control the protein function folding in the manufacture process. So actually in a biotechnology factory, you can produce the protein, but due to some technical errors, you may not have it properly folded, so no function for that protein. So it must the biotechnology uses that to understand. The process of glycosylation is a very important process for the function of protein. It's a post-translational modification wherein carbohydrate units are attached to specific location on proteins. More than 100 post-translational modifications occur within eukaryotic cells. This change can affect the protein activity by increasing its solubility, orient protein to the membrane, extend the active life of molecule in an organism. This glycoprotein, the spray protein undergo glycosylation, can be used to target and destroy B lymphoma cancer cells. The glycoprotein is combined with a nanoparticle loaded as a chemotherapy drug. Protein engineering by directed molecular evolution. It is the introducing of a specific predefined alteration in the amino acid sequence through a process called directed molecular evolution technology. It can induce mutation randomly into gene and then select the bacteria with the protein product enzyme that have the high activity. That's meaning you do mutation, as we said before, single nucleotide mutation, and you check if this mutation will increase the activity of that protein. Example, a company was able to produce bacteria and the industrial enzyme that tolerate high cyanide concentration that normally kill bacteria. Resulting selected bacteria can be used to remediate cyanide contamination resulting from mining and other waste accumulation. The protein engineering provides the possibility to invent proteins that are tailored or designed for a specific application. Example, using biotechnology to form synthetic infectious protein particle to study the following diseases. The faulty protein folding is expressed as a disease caused by infectious protein called brines, PRPC, which attract normal cell protein and induce a change in their structure, leading to the accumulation of useless protein that actually damages the cell. So brines can occur in sheep and goats and cows and causes mad cow disease. In human, it causes another a similar type of disease named according to the two scientists who discovered Creutzfeldt and Jakob disease, CGD, and this is a fatal degenerative brain disorder. The third subtopic is protein production. There are two major phases used in producing proteins, either upstream processing or downstream processing. Upstream includes the actual expression of a protein in cell, while the downstream involves the purification and verification of the function of the protein. In the upstream, we will talk about first using bacteria in upstream processing. Most common bacteria species used is the E. coli. They cultured in large quantity in a short period of time. They're relatively easy to alter genetically and can increase the level of production of bacterial protein by introducing additional copies of relevant gene. So you can introduce in the bacteria not single copy of a gene, you can introduce a multiple copy of genes, so to make sure to have a multiple copies of protein translated. The majority of proteins synthesized naturally by E. coli are intracellular. Most cases resultant foreign protein accumulate in the cells in the bacterial cytoplasm in the form of insoluble clumps, which are called inclusion bodies, which must be purified from other protein. Sometimes the genetically engineered E. coli produce the desired protein in the form of a fusion protein. That means it's attached to another protein. So the target protein is fused to bacterial protein, so an additional step is necessary to break these two parts. Fused bacterial protein is usually an enzyme that will bind to its substrate and can be attached to a purification column. So you use this step to purify the protein by attaching it by engineering and attaching it to another protein that used to purify. There are cons to expressing proteins in bacteria. Why? Because protein expression upstream. So we're talking about now about the fungi, using the fungi as in upstream processing. So it's we can use that source of a wide range of proteins used in products including animal feed and beer. Many species of fungi are used as a host for engineering products. These fungi are eukaryotic and capable of doing post-translational modification to allow proper folding of the proteins. You can see from this table, it's a knowledge table, some recombinant protein that is being produced from different types of fungi. If we use the plant as an upstream processing, plants can be genetically engineered to produce a specific protein that do not occur naturally. Tobacco plant is a great choice for biotech protein production. The disadvantage using a plant, not all protein can be expressed in plant. They have a cell wall which makes purification more difficult. The process of glycosylation is highly different from animal cell. We'll use mammalian cells as upstream processing. Actually, the nutritional requirement for a mammalian cell is quite complex. The mammalian cell grows very slowly and is very challenging. They can be easily contaminated. It is the best choice for protein designed to be used for humans. If we use animal bioreactor as upstream processing, we can use a monoclonal antibody production. Mice are injected with an antigen and the mouse secrete the desired antibody, and this antibody is then purified. If we use an insect system as upstream processing, the most famous is the baculovirus, which is the virus which infected the insects, are used as a vehicle to insert the mammalian DNA, causing the desired protein to be produced by insect cells. Sometimes the post-translation modification of proteins is slightly different in insects than in mammals. Currently, this technology is used for small quantities of protein which are needed for research. Let's talk about the downstream processing methods. Actually, we're talking about the protein purification. Of course, the step one is preparing the extract for purification. Protein must be harvested from whatever organism you used. The entire cell is harvested if the protein is intracellular, and this requires lysing the cell to disrupt the cell wall or the plasma membrane and release the proteins. The method include freeze-thaw, and the detergents, mechanical methods, mixture to release the content of the cells. After cells are ruptured, organic alcohol and salts are added to the mixture. The culture media is collected as well if the protein is extracellular. So when you are purifying extracellular protein, you need the cell culture medium, and this protein will be extracted from the culture media. The step two, after you purify, you need to stabilize the protein in solution. You maintain low temperature and a proper pH in the buffer solution. You add protease inhibitor and antimicrobials to prevent folding, protein misfolding, and to prevent protein from being digested. You add additives to prevent foaming and shearing, which will lead to destroying of the protein. These additive must be removed later during the purification process. Step 3, separating component in extract, because actually you are separating all the protein that been inside the cell, so you need to separate your own protein of interest. The method used for protein separation, first method is protein precipitation. So protein quite often have a hydrophilic amino acid on their surface that attract and interact with water molecule. This characteristic is used as a basis for separation of protein from other substance in the extract. Salts like ammonium sulfate are added to mix to precipitate the proteins. Solvents such as ethanol, isopropanol, acetone, diethyl ether can also cause protein precipitation by removing water from between the protein molecules. The method used of separation of protein, another method is the size-based filtration separation. Actually, you do centrifugation, that's another method second to protein precipitation. You do centrifugation, then you do membrane filtration, then you do dialysis. The centrifugation, of course, separates samples by spinning down at high speed. The protein can be isolated in single layer or separated from the heavier cell component. After that, in membrane filtration, thin membrane of nylon with varying pore size are used to filter out all the cellular debris. It's a faster method than in this slide, we can explain how we purify. So you can see this is a centrifuge. You have this centrifuge with centrifuging the sample. Then you have the cell homogenate before centrifugation and after centrifugation. You can see that the pellet is the larger and denser component sink in the bottom of the tube, while the supernatant contain the less dense components. This first one is small volume fixed angle centrifugation, while the other one, if you need to do a batch centrifugation, that you mean you have larger samples, and the centrifuge is equipped to collect from all the samples the proteins. You feed the samples, you feed the centrifuge with the cell homogenate, and then you can collect the protein supernatant. Continuing the size-based filtration separation, we'll talk about dialysis. Dialysis mainly rely on chemical concept of equilibrium. It is the migration of the dissolved substance from areas of high concentration to areas of low concentration. They used to remove smaller solvents, and other additives used in very early processes of protein purification. Salts are replaced with buffering agents to stabilize the protein during rest of protein to stabilize the proteins during the rest of protein purification process. And you can see from the figure, initially you start with a protein in a high salt protein solution, that you transfer this solution into a dialysis bag, as you can see, which have pores, and this dialysis bag is placed in a low salt buffer. In this case, you can see that the ions, after several changes of buffer, the salt diffuses out of the bag, leaving the new buffer, which goes inside the bag, and that's why we change the salt concentration of the protein. Continuing, step 3 is separating component of extract. We'll use the other methods is the chromatography for protein purification. So actually, it's a way to sort a protein by size or by how they bind to be separated from other substance. You have long glass tubes are filled with resin beads and buffer. This protein extract is added and flow through the resin beads. Depending on the resin used, protein either stick to the beads or pass through glass column, while beads acts as a filtration system. The other method is size exclusion chromatography. You can use gel beads as filtering system, so large protein molecule they will move around gel beads, while small protein molecule pass through more slowly because they are unable to enter the gel beads, which retard their flow. The other one is ion exchange chromatography for protein purification. This is depend actually on the electrostatic charge to bind protein to resin beads in the column. When charged proteins bind to the resin, the contaminants pass through and out of the column. So protein can then be released from the column by changing the electrostatic charge by increasing salt concentration of salt solutions. Anion exchange resin is positive charge, while cation exchange resin are negative charge. And you can see here from the figure how size exclusion chromatography work. You initially start with a low molecular weight protein, low molecular weight compounds and high molecular weight compounds. The one which is succeeded to move and got eluted from the bottom is the high molecular compound, while the others, the small ones, are retarded, so you can separate both compound from each other. And in this figure, you can see how you can measure the absorbance of the eluted compound based on the UV. You can see that the high molecular weight compounds eluted very early than the low molecular weight compounds, and you still can have some intermediate molecular weight compounds during elution. In this one, you can see the complete the ion exchange figure showing the ion exchange chromatography. So here you can see that you are taking a positive exchange column, and you have ions that is moving which have different charge, and you can see that protein X eluted first, then protein Y, then protein Z. Actually, you have a positively charged in the figure C. You can see that the beads are positively charged ion exchange beads, and they will attract more the negatively charged proteins. So you can see here at the bottom figure B, you can see the time at which the compounds have been eluted based on the time and based on the charge. We'll talk about the affinity chromatography as well used for protein purification. It actually depends on the ability of most protein to bind specifically and reversibly to ligand. After protein have bound to resin, buffer solution is used to wash out unbound molecules. Then special buffers are used to break ligand bonds of the retained protein, quite often used for purification of fusion protein. These techniques shortens the purification process by reducing the number of steps. So you can see here, you have the column resin bead attached to it the affinity ligand, and you have the protein ligand that will bind to this affinity ligand. So you can see here on the figure, the top one, you can see that the column resin with the column resin beads with the hydrophobic ligand, and you have the protein of interest with hydrophobic batches on its surface. You can see that this protein will bind to the ligand, okay? This binding to the ligand, after that, and the other stuff will pass through the column. After that, protein are eluted in order of increasing of hydrophobicity by linearly decreasing the salt concentration, and you can have here the peaks for different products have been eluted. Another method is hydrophobic interaction chromatography. Actually, proteins are sorted or isolated on the basis of their repulsion to water. Column beads are coated with hydrophobic molecules, and the hydrophobic amino acids in the proteins are attracted to similar chemicals in the beads. The other method is isoelectric focusing. Each protein has a specific number of charged amino acids on its surface in specific places, or due to this unique combination of charged groups, each protein has a unique electronic signature. We call it isoelectric point, where charges on protein match the pH of the solution. This behavior used to separate similar protein from each other. So basically, if you can have two protein of equal sizes, but they have different isoelectric point due to the difference of the charge on its surface. The other method is two-dimensional gel electrophoresis. It's actually a combination of isoelectric focusing and gel electrophoresis. First, you do first dimension of 2D gel is used to separate protein based on size. Then you do a 2D gel, you switch the gel exposed to electrical current, and now the proteins will be separated based on the charge. There are other analytical methods for protein purification. The most famous of it is HPLC, high performance liquid chromatography. They use a greater pressure to force the extract through the column in a shorter time than other methods of chromatography. The limitation, less protein is separated. You have as well mass spec or mass spectrometry. It's a very sensitive method used to identify small difference between protein, and they used on the outflow of the HPLC system to identify the proteins. The mass spec does three things. They suspend sample molecules into charged gas phase. They separate the molecule based on mass charge ratio charge by acceleration down in nanotube. They detect separated ions. The readout is produced, which indicates the identity and the size of most protein analyzed. The application of mass spec protein sequencing, to do this technique, first the protein is digested into smaller peptide fragment and analyzed by mass spec to determine the amino acid sequence. Protein verification. It is important to verify that the target protein is not lost during each stage of purification. So basically, first thing you do is SDS page. You can use a detergent. How it made its detergent, SDS, is added to the protein mixture, and mixture is heated. These sulfate charge are evenly distributed along the denatured protein, making separation dependent on size and not on the charge. After treatment with SDS protein sample, is loaded into BAGE, which is a polyacrylamide gel electrophoresis, where it forms a single band at a specific location on the gel depending on its molecular size and mass. So how you visualize the proteins after running on SDS page? The straightforward is to use a Comassi stain. It's a dye that combines with protein, so a color band results. Usually it's blue in color. You can compare the stain sample with known size and marker to estimate the protein size. During purification process, the colored band representing the protein should become increasingly intense, showing that its protein is not lost during the procedure. So in other words, during the purification procedure, if it's going okay, so that means during the purification you have an increasing, the band will show increasing color. Okay, so we can see that in the figure. This is the electrophoresis system used for electrophoresis, and you can see from the top figure, you can load a protein into the well. So this is how it goes when you run the sample. You can see you have 5 different stages of purification, and in lane 5 you have the desired protein only have been separated from the other component of the protein that came with the cell lysis of the organism. You can see this actually figure showing the protein have been isolated, and now it's clear in the sample. Another way of verification is the Western blotting, and it is a specific method for protein separation used after doing SDS-BH. So normally proteins after SDS-BH are transferred to nitrocellulose membrane using electrical current. All sites on the membrane are then blocked so that the antibody will not bind to them non-specifically. You put a primary antibody that is specific to the protein of interest and you incubate with the membrane. If there are any antibodies present that are directed against one or more of the blotted antigen proteins on the membrane, this antibody will bind to the protein and the other antibody will be washed away at the end of the incubation. That means if you got your antibody, if you got your protein of interest and you incubate with the primary antibody, it will bind. If there is no protein, the antibody will not bind. To detect the antibody that have been bound to the protein, a secondary antibody is used coupled with a reporter, which are usually alkaline phosphatases are added to the membrane. Excess secondary antibody as well is washed off the membrane. Then the substrate is added to this antibody, which is linked with alkaline phosphatase reporter group. Upon reaction, a signal, a visual band will appear indicating the presence of antibody. And you can see here, this is a readout of the Western blot. You can see that you have higher molecular weight than expected, often due to glycosylation. So glycosylation is the addition of carbohydrate group on the protein. Once the same protein have an additional carbohydrate group, it will migrate, it will be retarded because the migration is according to size. The third lane, it shows low molecular weight protein than expected, usually due to cleavage or degradation. So actually all of them, 1, 2, 3 are the same protein, but the normal one is in the lane 1. No, number 2 migrated slowly because it's glycosylated, so it's heavier in size, while the third is a little bit degraded, so it's smaller in size. The other method for protein verification is ELISA. It's a very sensitive, practical assay useful in detecting protein even at low concentration. You have a direct ELISA where the antigen or your protein of interest is coated on a glass or a membrane, and you have the primary antibody tagged with a reporter group that gives the signal once the substrate is added. You have indirect ELISA. Actually, in indirect, you need a secondary antibody conjugate with a reporter that gives the signal with a substrate. You have a sandwich ELISA. You need in this case, in sandwich ELISA, the scaffold is having antibody which is specific to your protein of interest, and then you use the primary antibody and then the secondary antibody conjugated to a substrate that gives signal, conjugated to a reporter that gives signal with the substrate. Then you have the competitive ELISA. That means you have the antigen or the protein of interest coated on the substrate, and then you incubate with 2 different antibodies. One is specific to your antibody and the other is not, and it only will give a signal when the right antibody binds to your protein of interest. So in all these 4 cases, giving the resultant signal is indicative of the presence of your protein of interest. So after you verify protein, you need to preserve protein. Once the protein of interest is isolated, collected, purified, it must be saved to preserve its activity. It can be lyophilized. Freeze drying is the best choice because it maintains the protein structure. The post-purification analysis methods include protein sequencing, of course, to know the sequence, mass spec, of course, to know the unique signature and what is this protein. The methods include as well X-ray crystallography. This used to determine the tertiary and quaternary structure of protein, and this requires a pure crystal of protein that have been dehydrated from solutions. When exposed to X-ray, a pure protein creates a specific shadow pattern based on its configuration. So using a computer analysis of the pattern allows the description of the structure of proteins. The fourth subtopic is proteomics. Actually, proteomics is a scientific discipline dedicated to understanding the complex relationship of disease and proteins expression. Proteomics are compared between healthy and diseased states. The variation of the protein expression are correlated to the onset of progression of disease. The goal is to discover protein markers to be used as a diagnostic method and development of a target therapy or target drug. The techniques used for proteomics, it's a two-dimensional gel electrophoresis separates a protein followed by a mass spec to verify the protein identity. They can use amino acid sequence to characterize the protein. Protein microarrays can be used as well to set a protein immobilized on a surface coated with a reagent that will indicate binding by color change. Functional protein microarrays have been recently applied to discovery of protein interaction, protein carbohydrate, protein lipid, protein DNA, protein drug, and protein-protein interaction. Thank you.